Okay, so welcome to the Genetics Lab. So the Arabidopsis project, the goal of the project is to identify the function of an unknown gene. So we have done already BLAST to find any orthologs in other species, and also to see if there are any specific domains, conserved domains. And last week we have done EFP to see where the gene is expressed. So this week we will do another tool which is called Expression Angular, and that mainly helps us find correlated genes that have similar expression pattern like yours. So it would be really useful to find genes that have the same expression pattern like yours. So let's talk about correlation and what that means. So two variables are said to be correlated, right? If there's a specific pattern, if they change in the same way. So these two variables, they go together and they have an R value or correlation coefficient value of 1. So they're perfectly correlated. These ones are still positively correlated, 0.9. It's kind of a little less. And here, there's no correlation, right? So any change in X does not predict the change in Y. And these two variables are negatively correlated, negative 0.5, negative 0.9. And these two variables, uh, they have a perfect negative correlation. So it's the value of its negative 1. So, and when we say that two variables are correlated, it does not mean that they have equal value, right? The values are actually different, but the trend or the pattern is similar. So if you look at this, for example, like these are the change in years, and this is the human height over years. So there's a trend, like the female height, this is the male height, this is the average, the national average. So you can see that the x-axis, the years goes from 1920 up to 1996, right? So this is 1996. And on the y-axis is the height is in centimeters, right? 175. So the x and y, they are not equal, right? They don't have to have equal values, but they should show a similar trend. Similarly, if we wanted to talk about genes, so let's talk about genes. So this is gene X and this is gene Y, right? On the x-axis is the expression in different tissues, right? Maybe leaf, roots, like different tissues. On the y-axis is the actual expression value, the fluorescence that you see when a gene is expressed. Gene Y, the same, the different tissues over here could be different days, and on the y-axis is the expression value, how much of the gene is expressed. Are genes X and Y, are these two genes, do they have a similar pattern? Well, at the first blush, when you look and you kind of see that this is really low, this is really high, you might be inclined to think that they're not really correlated. But if you look a little deeper, you will see that these two genes, like this one, it peaks at 2, 6, 10, and 14, right? Similarly, this one peaks at 2, 6, 10, and 14. So they are actually similar in their pattern, right? Although the values are not really the same, but they follow the same pattern. So maybe that gene is not generally expressed in higher doses, right? And as compared to this one. But that doesn't mean that they're not correlated. They are actually highly correlated. So what this week's tool will do, which is Expression Angular, it takes your gene and calculates the correlation coefficient, right? with all other genes in the genome, right? It does a pairwise comparison with every single gene, like the 23,000 other genes. So, and it calculates, right, the average correlation of all other genes. So, so this is like the equation for most, like for mathematically inclined folks. So the expression, and x is the mean of the expression for gene X, and this is the standard deviation for that gene expression. Oh, let me go this here. And this is again gene Y that we're testing, and this is the average of gene Y divided by the standard deviation. Anyways, it gives you an R value. So for each gene, it kind of looks at the expression for all the tissues, all times, under all conditions for the same gene. So this is my gene. It tests that, it compares it to gene Y under all conditions, under all tissues, under all abiotic stresses, all biotic stresses. So, and it gives you a value, okay? And it tells you how these two genes, how their pattern matches, okay? So let's look at this week's tool. This is how the output will look. So let's look at one of those. Let's do one of these analysis. So this is the link that you guys have. This is expression Angler. So this is the link that you guys have. This is the link that you'll click, and it takes you to this website. And let me put this gene, unknown gene. And you can see, put the gene name here, we'll ask it to return the top 25 genes, the top 25 genes that are closely related to my gene, that their expression matches mine. So, and you would see that there are multiple, there are different sources, different databases, like 10, okay? So, and I'll ask it, I'll just submit query. And I will click formatted data after median centering and normalization. And this is the figure, how it looks. Let me make this a little smaller. So, and you can see that, I mean, this figure, it shows you, I mean, there's something going on, right? It's really highly expressed to the right and not that much to the left. So, and we'll talk about this in a second. But here it shows you, these are the 25 genes that are highly correlated with your gene. And if you look here, this is like the correlation coefficient value. So here is, it's actually the perfect, so this is the same gene with itself. How is it correlated to itself? You can see that this number here is 1. It means perfect correlation with itself, right? I move down here to this gene, and you see that the number up there have changed to 96, right? Look at this numbers when I'm moving. So 96, 96, so all of those genes are highly correlated. So two things we want to look at here. So this figure, it looks like it's really highly expressed in here, in this right part of the figure. And what that tells me, well, you could see that tissue right here, all of this green thing, let me make this small, this is actually the shoot, right? It tells you that this is the shoot. So all of these tissues, all of these are samples that have been collected in the shoot. Smaller. Okay. So the shoot, and the ones to the left in here, this is actually the root, which is this. So for each given sample, right, it tells you which tissue, what tissues that at, and tells you which growth stage, right? It could be, I mean, these are so the growth stages, it tells you right here. You could find out what those mean. So, but it also tells you the treatment. So the black is the treatment and the white is the control. And this one tells you the time, like how many hours after treatment. But anyways, the way I would interpret this figure is that, okay, it's highly expressed in this side of the graph, which is mainly the shoot, like the tissue, which is the shoot, right? So all of these genes, they're expressed together in the shoot. So what functions or what genes can be highly expressed in the shoots? Well, one thing could be the shoots are the leaves and the stems, the above ground math, right? And they're known to be doing photosynthesis. So another thing you could look at in this figure is this chart to the right, and each bar of these kind of denotes a specific function. So maybe gene number, like this gene, right? It has a bunch of cellular functions, right? Each bar, each color denotes a certain function. But one color that I see common across all of these genes, right, is this green bar, right? All of them, almost all of them have the green bar. So I go down here and try to figure out what the green bar is. I go down here and you see that this green bar, right, it says chloroplast. It means that these genes are kind of correlated. They have something to do with the chloroplast, which is the photosynthetic machinery. So, so this figure would be interesting, right? I'd be interested in analyzing this one. And exactly, so you don't necessarily have to interpret all of these, but in this case, I see that I'm more interested in the tissue because it is what gives me more differential expression, right? For example, if you saw that, like this one, for example, is highly expressed, this was not highly expressed, like this yellow line, let's look and see what that yellow is. So I think yellow is here. I could hover over this one and move from left to right. And it tells you that this is osmotic stress. Let me move up. So this tells you that this is the time. So the darker it means more time after treatment. So if I move to the left or right, look at this one, the type of stress, and also the growth stage and the time in hours. Look at these, they will be changing. So I move to left to right. So this is after 12 hours, after 24 hours of treatment. It's not really expressed. You could say that it's under osmotic stress. It's not really highly expressed. So this would be one interesting figure, right? So let me go back and do an analysis about stress. Let me pick maybe the seed compendium. Let me click seeds. This. Okay. Seeds. So, okay, let's see what that means. So this is kind of not really, kind of a little down regulated. So what is this? Why is this color different? So let's see what is that, what that blue is. So that's the mutant. It's downly regulated in the mutant, and that's in the wild type. So I see it's down regulated in the mutant. Let's see, so all of this is, so this is the mutant that's called WS. It says here WS. This is one type of mutant. So why is, you can ask the question, why is my gene downregulated and it's not real in that specific mutant? You can ask that question. So, and again, you see that a lot of the genes that are correlated here, most of them have that photosynthetic job. They have something to do in photosynthesis. So, and again, as a rule of thumb, if the correlation is below 0.7, right, then that's not really that interesting of a correlation. So Let's. So you don't really have to consider all of the 10 databases. You should explore all the 10, but just pick one or two that are very interesting. So, for example, if your gene based on the EFP from last week, you have noticed that your gene is highly expressed in the roots, then you'll probably want to also look at the roots and see how it looks over here. Okay, so let's see this. Okay, so this is the root. I see that my gene is kind of highly expressed in here. So I want to see what is that? What is that? So this is the root. It's highly expressed in here. So let me see what that part area is. So you could see at the growth stage, you could see that this area to the left, this is actually the whole. Okay, this is the root, lateral root, whole root. I think tissue let me. So these are the growth stage. So what's common across all these tissues is that they're all the same growth stage, which is 1.2, which is right here. So 1.2 means we can look here. 1.2 talks about the leaves. Is it 1.2 or 1.0? 1.02. It means second rosetta leaves that are greater than 1 millimeter in length. So the rosetta leaves, like those leaves in Arabidopsis, the bottom leaves. So this tells you that they did the analysis of the roots, but this was at the early stage, right? When the Arabidopsis has just was at this stage, at the early rosetta leaves, 1.02. So this is the expression roots early on. Okay. And you can actually, if you see something interesting over here, maybe there's like a red line, you could ask, what is this? Right, you could look over here and it tells you what this is the whole root. And also this was spill specific nitrogen response in Arabidopsis. And let me go up here, see if what changes over here. So this is the actual tissue, right? Tells you what the tissue is. This is the whole root, and this actually here tells you if there is the last row shows you whether there was a treatment or no under a specific treatment. And actually here, these are the colors of the different tissues. You could actually find out like the specific one. This is, if we're interested in this column, you could look at this brown and it should tell you what type of tissue that is. This is the whole plant, but let's see if there is anything more specific here. Right, that's the whole root. Okay. You could see if you could extract any type of information from this. But again, you don't really have to include all the 10 databases. Just pick the top, like the ones that give you the best data. So let's look at if we have picked abiotic stress. So you can also find the genes that are negatively correlated, right? Why would you pick the genes that are negatively correlated? Well, if a gene is negatively correlated with your gene, meaning when your gene increases, right, that other gene expression decreases, or vice versa. So that could be that this gene is like a negative regulator of your gene, right? So to find those negatively correlated genes, I would just pick bottom 25 genes and just hit submit. And that should show you like the genes that are negatively correlated with your genes. And again, you hover, like this is the correlation coefficient, you hover here and you see that this value changes negative 0.9 to negative 9. So all of these genes are negatively correlated with your gene. When your gene is highly expressed, these genes are downregulated. So you can ask, why are those genes negatively correlated with my genes? And again, you can look at these bars, and you can also hover over like the gene names, right, and see exactly what they are doing. That could give you a hint. So if I hover over here, it shows you like the name of the gene here. So let's move down. This is GM, gram domain family protein. I don't know what that does. Isocitrate dehydrogenase, metallothionein, oxaloglutarate. So you can see if any of these genes, I wouldn't really dig deeper into every single one of them, but I would hover over these genes in case you see like something specific or something in common about all of them, right? If all of them have the name dehydrogenase in them. Well, for example, this one has dehydrogenase, oxygenase, glutamate synthase. They're kind of different names, right? So that's not very helpful. So, but I think it might be useful to look at the gene names, especially with the positively correlated genes. It kind of tells you something. So let's see these genes. Let me click this. So let's move over these genes. So this says the name of this gene, Rubisco methyltransferase. Oh, that's my gene. So the other one says tetrapeptide. Know what that is. So the next one says photosystem 2. Photosystem 2, this is part of photosynthesis, right? The next one, it says PDE. At the end of the line, it says chloroplast. The next one, CURT1, you can check what that is. Peptide, okay. So there are actually this one, superfamily protein, ascorbate peroxidase. Yeah, their names are not really, didn't really help, but okay, this one says plastid transcriptionally active. So it seems like we have a few. This one too, it says PERT1B, but then it says photosystem. It has also to do with photosynthesis. Okay, so just do a little bit of digging and look at these genes and see if you can find something useful. So this is an example of one of the genes. So this is how the output of the gene looks, right? This is the number of the genes, like the experiment identifier. This tells you the experiment category, what type of research area that is. Again, this is the growth stage. So the darker, it means the older plants. And this tells you which tissue of the plant that is. Then if there are specific treatments, so the black would be a treatment and white would be control under certain conditions. So you could read this as this is one of the experiments, and this was at a certain growth stage in the leaves, and this was the treatment, right? And this was the time, how many hours after the treatment, and this if this was the mutant, okay? So, and these are kind of the functional bars that tells you what the function is. So let me show an example of one of the so this is one of like an example of one of the genes. This is one of the genes that we found. In this case, like this figure looks interesting, right? You could ask, okay, this says that it was highly expressed in the root. So this is the shoot, this is the root, right? The root, and specifically under what type of stress? You go under treatment, here, and you would know that this is specifically salt stress, right? So you look at the correlated genes and you see that they all have that brown bar, which tells you it's in the nucleus. So you could conclude that your gene is highly expressed under salt stress, and all of the genes that correlate with it are kind of expressed in the nucleus, localized to the nucleus. We can ask, are they maybe transcription factors that respond to salt stress, or what exactly are they? Okay, so there's one example of something interesting that you might see. Another unknown gene we looked at, this is the one that I've just discussed earlier. And these bars tells you where it's expressed. So you could conclude that other genes that have to do with photosynthesis, photosystem, phototropin, rubisco, all of these are known to be involved in photosynthesis. Another gene this is another unknown gene that we looked at, and you see that it's kind of highly expressed in here. You could look and ask, what is this? When you hover over with the mouse over this, it tells you which tissue, where it was expressed. So, and we also talked about doing the negative correlation. Check the negative correlation, okay? So I think I'll just leave it here. And so again, you don't really have to make an extensive list of all the genes or of all the hypotheses that are possible. Just pick one or two and just kind of follow on these. Okay, well, thank you, and I will see you next week.